Assalamu alaikum. I would like all of you to please be seated so that we can start our conference. Kindly be seated. Assalamu alaikum. I would request all the people present in the hall to be seated so that we can start our conference. Bismillahirrahmanirrahim. Welcome to the Pakistan Society for Rheumatology Annual International Conference. Let us begin with the name of Allah by hearing a few verses from the Holy Quran.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم الحمد لله رب العالمين الرحمن الرحيم مالك يوم الدين إياك نعبد وإياك نستعين اهدنا الصراط المستقيم صراط الذين أنعمت عليهم غير المغضوب عليهم ولا الضالين
Ameen. Jazakallah. Kindly, I would request you all to stand up for the national anthem. Thank you. So let me begin by introducing myself. I am Dr. Saira Elaine Anwar Khan, and I work as Head of Department of Rheumatology at Shalimar Medical and Dental College. I'm joined by Dr. Bilal Azim Bhatt, who is Head of Department of Fatma Jinnah Medical University. Both of us are humbled by the Pakistan Society, who thought that we were worthy enough to be able to convene this conference. Pakistan Society of Rheumatology was formed in 1995 by a handful of doctors from the UK and USA who had returned to their homeland to serve the country. We have now reached 25 years since its inception, and we are proud to be celebrating the Silver Jubilee conference. None of this had been possible had these leaders not returned to their homeland and served the country. I thank them for their contribution towards the Pakistan Society and for the country at large. During this conference, there will be multiple scientific sessions and multidisciplinary team meetings. In these meetings, We will be discussing all aspects of rheumatology from speakers who are well known across the country as well as all across the world. We thank them for their contribution and time and commitment to be doing all these activities for the betterment of our learning experience. I also thank my patron, Professor Nigat Mir Ahmed, Chair Scientific Committee, Professor Sumaira Farman Raja, and Professor Dr. Ahmed Saeed, without whose guidance none of this would have been possible. So without much ado, I will begin the conference by handing over the stage to the stage committee, Hadia and Hadi, and they will take over from now. Thank you very much.
Greetings and assalamu alaikum. My name is Hadi Amyan, and I'm a second-year MBBS student from Shalimar Medical and Dental College.
And my name is Dr. Hadiul Hassan Khan, and I'm a second-year MBBS student at Shalimar Medical and Dental College.
And we will be mediating the first session. Before we proceed, I'd like to ask everyone to please turn off their mobile phones to avoid any disturbances. If you have any questions, you may write them on the question cards that are being handed out by the hall committee, and they will be answered at the end of the session. Please also remember to fill the feedback forms provided to you by the hall committee.
The conference will begin with our first session on lupus and APLS. And I'm honored to announce that this session is dedicated to the late Dr. Azra Arif Ali, who served as the very first president at the Pakistan Society for Rheumatology. From 1995 to 1997, Dr. Azra was a consultant rheumatologist at the Aga Khan University Hospital in Karachi, where she pioneered advancement in the field of rheumatology in Pakistan. And we are honored to dedicate the first session in the Silver Jubilee Conference to her.
Before we begin, let us introduce our honorable chairs for session 1. We are honored to have Professor Dr. Samina Ghaznavi, who is the current president of the Pakistan Society for Rheumatology. Will Dr. Samina please come onto stage? Should I say it?
Our first speaker today is Dr. Sarfaraz Hasni, who is the Director of Lupus Clinical Research as well as an Associate Research Physician at the National Institute of Arthritis, Musculoskeletal, and Skin Diseases in the United States. Having significantly advanced clinical research in SLE, I'd like to invite Dr. Sarfaraz Hasni to speak on untangling the knots in complex SLE pathophysiology.
Working now? Okay. So, good morning and assalamu alaikum to everyone. And first of all, I would like to begin with thanking the organizers of this Silver Jubilee 25th anniversary of the Pakistan Society of Rheumatology conferences. I'm really honored and humbled to be here among friends and to this very momentous occasion on the map of the rheumatology in Pakistan. And of course, very happy and excited to be back in Lahore, my second time in Lahore, and I really enjoyed the hospitality and of course, the accommodations done by the committee here. So thank you again to everyone. So these are my disclaimers, and of course, what I'm going to present is mostly my personal views, and these are not reflected of the institute that I work at. And these are the learning objectives of today's talk. So let's just begin with this slide, which is a review recently from George Sokas. And George Sokas has shown here that how complex the pathophysiology of the lupus is. And as you look at the top 3 blue bars here, it looks like that the 3 major components that sort of incite the dysregulated immune system are related with genetics as well as some environmental factors, and then there is, of course, a hormonal dose which does interplay. And all these 3 overlaps. And when these 3 events come together, then they lead to a dysregulation of the immune system. And the dysregulation of the immune system is in both parts, both the innate part of the immune system as well as the adaptive part of the immune system. And once this dysregulation starts, then it kind of goes back and forth and goes into a feedback loop where the adaptive immune system further perpetuates the dysregulation of the innate system and vice versa. And this all culminates into formation of the autoantibodies, and the autoantibodies then combining with the autoantigen forming the immune complexes. And these immune complexes, when deposited in various organs such as joints, skin, kidney, and so forth, lead to the clinical manifestations of the lupus. So it's So it's fair to say that the pathophysiology of the lupus is very tangled up, as you can see there, and it's no single pathway can explain the underlying pathophysiology of the lupus. And this is something which is, we have to kind of think about it in different ways, whether the innate system or the adaptive system or both together working to cause this dysregulation. So how do we untangle this tangled up mess? So before we even go there, I do want to present to you sort of a framework about how to think about lupus. So lupus, as we have just mentioned, that has a background of genetic factors, risk factors, in a patient who is susceptible to autoimmune diseases. And then some stochastic environmental event happen, for example, an infection or some other environmental exposure or chemical exposure that leads to immune and molecular heterogeneity. And this leads to a variegated clinical response, which most of the rheumatologists and non-rheumatologists are very well aware that the lupus is a very heterogeneous disease in clinical presentation. But I think the point that I want to impress is that not only it's heterogeneous in clinical presentation, the heterogeneity is extended further down in the immune dysregulation underlying the pathophysiology of the lupus. So you can think about lupus as a single entity where all of these factors come together, and lupus is somewhat, as one of the sessions will show, that has multiple faces. So we can think about it as one disease with multiple heterogeneity, or we can think about it as a disease which has multiple, as different diseases which have some common threads underlying the immune pathophysiology and clinical manifestations. So these are 2 ways of thinking about lupus, and maybe if we sort of keep that framework in the mind, we can somehow try to untangle the underlying pathophysiology of this complex immune pathophysiological mechanism. So I'm going to present to you 4 studies to try to untangle, and there is a common theme through all these 4 studies, which I will come to the end, so I'm going to keep that a little bit of a surprise. And then, in addition to that, there will be 2 other additional mechanisms that I'll be talking about. So this first study from Virginia Pascal's group, which was published a few years back, where they looked at the clinical and transcriptional profiling of 158 pediatric lupus patients, and then they followed them for almost 4 years. And during this time frame, they did have close to 1,000 unique visits. And during each visit, they collected the clinical information about these patients, which included their disease manifestation, the disease activity indices, as well as the various treatments that they were having. And then at the same time, they also collected blood samples from these patients. And then these blood samples, they did the transcriptic profiling, so looking at the gene expression data associated with all these 124 visits. And then they compared that to 48 healthy volunteers, which they have collected the clinical data as well as the blood samples along the same time. And then multiple statistical modeling were done. And of course, this is a very complex and big paper. And of course, there's not enough time to discuss all the different nuance of this paper. But just to summarize this, the key finding from this paper showed that the prevalent interferon signature was present in most of these samples. So close to 85% of the patients' samples did have a gene signature of interferon. And you can see here, these are the healthy volunteers, and these are the heat map of patients who have lupus. And on the very top, that red dotted line, are all the interferon stimulated genes. And as you can see that most of the lupus patients have an overexpression of these genes in their blood samples, and the patients who have healthy volunteers do not have that. In addition to looking at this, they also did the immunophenotyping, and the immunophenotyping showed that the plasmablasts were most robustly present in patients with disease activity. So when SLEDAI scores were high, the plasmablast levels were also high significantly, and this was more commonly seen in patients of African American ancestry as compared to the white and Hispanic patients. And furthermore, another important finding from this paper was that the neutrophils were perhaps for the first time, shown to be very active players in pathogenesis of lupus nephritis. And interestingly, all of this data, they have put it on publicly available website, which is WebSLE, and anyone who is interested to look at the raw data or do some more analysis, it's available for you at this website. So this was the first study. Now looking at the second study, this is the study from the AMP group, which is the Accelerated Medicine Partnership Group. And this AMP, just a quick background, is a partnership between the NIH as well as the pharmaceutical companies. And they both came together, and then they say that over a period of 5 years or so, they will invest in basic sciences to understand the pathophysiology of lupus. And with this partnership, the AMP group was formed, and the AMP group has published several papers, but I just chose one paper to highlight a specific point. And in this paper, what they did is that they looked at the immune cell landscape in kidney patients with lupus nephritis. And they have standardized the process of collecting the lupus kidney samples, as well as how they will process it, and all of that was processed at a central place. And then they did some statistical modeling and understanding how, which type of cells are involved in pathogenesis of lupus nephritis. So overall, they collected renal tissue, urine, and blood from 24 patients with active lupus nephritis, and they compared it with 10 controls which have kidney involvement, not secondary to lupus. So they have samples available, and then they compared them. And what they did is they did the single-cell RNA, single-cell sequencing of these immune cells. And interestingly, the important thing that was found from this was that the type 1 interferon were elevated in both blood as well as all the kidney cells. So single-cell profiling, as when they look at all the different immune cells, that was they were focusing on, and every cell has an increased expression of interferon genes. But most importantly, the B cells as well as the CD4 positive T cells had the highest level of interferon. As you can see from this picture, that both the highest gene expression was in the clones of cells who are B cells as well as the CD4, and these were the interferon gene score. It is present in pretty much everything, but this is the most highly expressed, these two type of cells. All right, so now I'll present to you the data from our own group, and as a PI of the Lupus Natural History Protocol at the NIH, which has been actively collecting samples for over 27 years now. We have a huge data set of both the clinical information as well as the blood samples. So over the last decade, what we have done is that we started collecting samples in a cohort of 130 of over 1,000 patients, and we also have collected samples for 100 healthy controls. And this is a longitudinal follow-up data of about 6 years. And during this 6 years time period, we have collected the clinical characteristics of these patients, which include the various treatments that these patients were undergoing, as well as the disease activity measures, as well as the damage accrual measures as depicted by SLEDAI and SLIC respectively. In addition to that, what we did is that we looked at a very broad profiling of these patients. So in the previous paper by Virginia Pascal and the AMP group, they were only looking at the immunophenotyping as well as the gene expression data. Whereas when we did this, we did one more thing, because our understanding was that the gene expression is there, but the gene expression may not completely explain what is going on in the blood. So in addition to the gene expression, we also wanted to look at the proteins which are formed by the expression of this gene and which are circulating in the blood. So in addition to the immune cell phenotyping by flow cytometry, gene expression data by RNA sequencing and nanostring, and then we also added the proteomics data to this. And interestingly, it's a huge paper, and we have submitted for publication. It's not published yet, so I'm not going to go through all of the data here, but I can just focus on the proteomics part of it, that the 3 clusters of proteins were found to be very highly expressed in the immune cells. And these 3 clusters, 2 of them were, as expected, were the interferon expressed as well as the granulocyte-associated proteins. And these correlated not only with the gene expression. So again, in the previous study, they only showed the gene expression, but we were able to show that not only the genes are expressed, the proteins are also expressed, and those proteins are interferon and the granulocyte associated, which is basically coming from the neutrophils. And in addition to that, we also were able to find out that certain specific proteins are associated with certain clinical manifestations. So for example, the interferon was typically associated with rash, and some of you who are familiar with the anifrolumab study data will concur that anifrolumab was mostly effective in patients who have skin manifestations. So skin manifestations seems to be clearly associated with a high interferon presence in the blood. And the granulocyte actually was more present in patients with arthritis, proteinuria, and lupus nephritis. And if you recall the data from Virginia Pascal's group, that they show the active cells in lupus nephritis are mostly neutrophils. So again, we are showing that the proteins which are coming through the granulocyte expression are mostly associated with lupus nephritis. But in addition to that, we also were able to identify a unique set of proteins which were not previously shown to be present in the samples of lupus patients. And these proteins are expressed by the APCs, or the antigen presenting cells. And most of these proteins, and this protein signature was associated with renal disease. And also, because we have this longitudinal data for almost 6 to 7 years, this presence of this protein expression at the beginning of the data was predictive of the future damage accrual, especially secondary to renal disease. So this, we are very excited about this data, and it will soon be published, and I should be able to, to share more data from that. And this is just the forest plot showing the ICP was most highly significantly associated with the damage accrual. Okay, so now after these 3 studies, I will present to you the data from the 4th group, which is Judith James' group at Oklahoma Medical Research Foundation. And they have previously shown that in the pre-clinical stage of lupus, before somebody gets diagnosed with lupus, there is a high presence of autoantibodies. And the high presence of these autoantibodies can be present for several years before a patient gets diagnosed with lupus. And the reason they were able to do that is they have access to the Department of Defense biorepository, where samples were collected from all the recruits coming to the military and has been biobanked. And they went back and see the patients who were subsequently diagnosed with lupus. They went back in time and looked at their sera and samples and see what were the things that were present in their serum 3, 4, 5, 6 years before they were diagnosed with lupus. So the autoantibody story has been presented several times, and I'm sure most of you are already familiar with this. But this is more interesting, that even before the autoantibody, for example, the double-stranded DNA by this mustard or gold line, which is coming very close to the time where the time 0 where they were diagnosed with lupus here. So it's not as high, but it's present there. But more interestingly, the 2 cytokines, which are IL-5 and IL-6, are present several years before the presence of the autoantibodies. And these cytokines are pro-inflammatory cytokines are present in very high concentration. So 2 points that I want to make from this is, one is that in terms of predictive modeling, if we want to predict who will in future develop lupus, if we combine the levels of IL-5, IL-6 with presence of the autoantibody, this is found to be the most predictive of somebody who will develop lupus. Because as you get consulted for patients who have positive ANA, but you don't know what to do with them. In the past, it was thought that if you longitudinally follow, eventually they will develop lupus. But we don't know how many of them develop lupus, because a vast majority of them will never develop lupus. But then if you combine that positive ANA, if you are able to detect, check IL-5 and IL-6 level, and if you combine all these three together, that has a very high predictive value of future development of lupus. The second point that I want to make from this is that the IL-5 and IL-6 are the cytokines which are effective in terms of activating the T helper or Th2 cells, as well as the Th17 cells. And because of that, it seems like that the inciting event before the lupus starts is the dysregulation of the T helper cells. And the T helper cells, when they get dysregulated, then they go and activate both the autoreactive B cells as well as the innate immune system. And all of this coming together can start to self-perpetuate. If you recall the first slide from George Sokas, that it does lead to this complex interaction between the innate and adaptive system, and eventually leading to the clinical manifestation of lupus. But things have been going on for several years before you get to this point where you diagnose somebody with lupus. Okay. So, so this is basically, again, a summary of what I was just talking about. The Th2 cells were the first to get dysregulated, and then they lead to interferon, and then they lead to autoantibody formation, and then the multiple immune systems immune pathways are dysregulated, and eventually this leads to tissue damage, and this is the time where patients are diagnosed with clinical lupus. So I just quickly want to talk about two other things, and I know the time is almost up, so let me just quickly go through this. So in addition to the innate and adaptive system, the two other concepts that have been very clearly shown to be involved in pathogenesis of lupus is the mitochondrial dysfunction, which has shown to be stimulating the abnormal immune cell metabolism, and as we all know, the mitochondria are involved with the oxidative phosphorylation. And the mitochondrial dysfunction leads to hyperpolarization and abnormal oxidative phosphorylation. So they are in a hyperactive state. So they keep on producing this oxidized phosphorylation, and leads to abnormally increased T cells and macrophage activation. And also there is a defective clearance of the apoptotic cells, which are the primary reason that we have autoantigen present in the blood. And then this excessive production of the reactive oxygen species, especially with the exposure to UV light, leads to this oxidative stress, leading to a pro-inflammatory cytokine milieu in the body. And this, in addition to that, the mitochondrial gene itself also shown to be associated with SLE pathogenesis. And interestingly, people have tried to target the mitochondrial dysfunction in lupus, and with some success, especially Andreas Perl has shown using the N-acetylcysteine in patients with lupus has shown to be associated with improved outcomes. The second concept is about the net, and this is something which is Marianne Kaplan from our group, she's my boss, has at the forefront of this research, where she has shown that the special population of the neutrophils called the low-density granulocyte, or LDG, lead to formation of netosis, where the chromatin network are extruded from the neutrophils, and it leads to exposure of the chromatin and DNA, as well as it also leads to formation of the proteolytic enzyme. And this has consequences in terms of increasing in inflammation, increasing the type 1 interferon production through plasmacytoid dendritic cells and the pro-inflammatory cytokines, and this is, as I said, as a potential source of autoantigen. And in addition to that, our group has shown that this may have role in the premature cardiovascular disease in patients with lupus. So I'm just going to skip this real quick. So in terms of summary, we can say that the genetic risk and environmental factors, some stochastic event happens, and as well as the hormones, they lead to increase activation of the innate immune system. The TLR7 activate the PD plasmacytoid dendritic cells. Netosis and LDG and mitochondrial dysfunction is also involved in the pathogenesis of lupus. And this all leads to abnormally functioning helper T cells, or Th2 and Th17 cells, as well as the type 1 interferon, and leading to B cell activation. And this leads to autoantibody production. And the autoantibody production then combines with the antigen and leads to the immune complex formation and complement activation, which leads to the tissue injury. So of course, this is an overly simplistic way of looking at the pathogenesis of a very complex disease. Again, it's fair to say that the untangling needs multiple arms, multiple people need to come together. And at the end, I do want to leave you with this thought, or in terms of this framework about how to untangle this knot. What can we do? And I think the four studies that I show had one common theme, that they were all relying on these big, large cohort that were prospectively collecting data, both clinical data as well as the biospecimen. And I think we are trying to establish the same thing in terms of Lahore Lupus Clinic, where we will be collecting data from a huge resource of patients, and then we will be able to maybe understand or contribute to the understanding of the pathophysiology of the lupus. So establishment of the large multi-ethnic lupus cohort, longitudinal collection of the standardized clinical data, as well as collection of the biospecimen and tissue, and using a multi-omics approach will be important, and doing something like whole exome sequencing, and then using some of the cool bioinformatics tool can perhaps help us understand or untangle this knot of complex pathology. And with that, I would like to thank you for your attention, and I'll be happy to answer any questions. Thank you.
Thank you, Dr. Sarfaraz, for the enlightening talk. Before we proceed, I'd like to take a minute to introduce Dr. Shamsa Humayun, who has just joined us. Professor Dr. Shamsa Humayun is a professor of gynecology at Fatima Jinnah Medical University, and we are extremely honored to have her with us today. Our next speaker is Dr. Laura Lewandowski, who is an assistant clinical investigator and the head of the Lupus Genomics and Global Health Disparities Unit at the National Institute of Arthritis, Musculoskeletal, and Skin Diseases in the United States. I'd like to welcome Dr. Lewandowski onto stage to speak on the genetics of early onset lupus.
Good morning, everyone. I'm Dr. Laura Lewandowski, and I am really excited to be here with you this morning. This is my first time ever visiting Pakistan, so I really would like to thank APLAR and the Pediatric Rheumatology Society for welcoming me to join you today. And I'm really excited to talk to you about the genetic landscape of childhood onset lupus. I have nothing to disclose, and all the views in this talk are my personal views and do not reflect those of the NIH. The goals of my talk are going to be to review the genetic landscape of lupus in global populations, discuss common variant studies and polygenic risk scores in childhood onset lupus, and review rare variants and their roles in pathogenesis of childhood onset lupus. So a little bit of my background. I have been studying lupus in populations around the world since I was a fellow in rheumatology. So during my fellowship, I did research in childhood onset lupus both in the US and in South Africa. And I lived in South Africa for 2 years studying lupus there. So I'm really interested in understanding more about lupus in global populations and in childhood onset lupus. And I'll talk to you a little bit more about that today. So as Dr. Hosny already reviewed, we know that lupus is a chronic multisystemic autoimmune disease, and the heterogeneity of the disease can often make it really difficult to study. But we know the inflammation can lead to irreversible damage and sometimes early death for these patients. Now, childhood onset SLE is a really severe form of the disease. So about 20% of all lupus patients are diagnosed during childhood. And children will have a higher disease activity and require more therapy than their adult counterparts. They're more likely to have the severe manifestations of lupus, like lupus nephritis and CNS lupus, and they accrue more damage than their adult counterparts, despite having younger, healthier bodies. And when I was living in South Africa, I found that this was true in these South African children that I studied while I was there. So I worked with a collaborator, Dr. Chris Scott, to collect a registry of more than 100 early onset lupus patients there. And just like in the USA, those patients had more severe phenotype than their adult counterparts. And in South Africa, those patients actually, I compared the patients in South Africa to a very large registry in North America, about 900 early onset lupus patients, and found patients in South Africa were even more severe than those in North America. So they had more disease activity, more severe organ damage, a much higher mortality rate than those in the USA. And part of that might be delayed diagnosis and access to care, but there were some patients in that cohort who also were diagnosed early and got treatment right away, and they just had raging lupus that raged on despite any medication that we gave them. And I've experienced those patients that just are recalcitrant to any treatment in the US and in South Africa. And so I really began to think that studying childhood onset lupus in diverse global populations is really important to understand more about this disease. Now, as Dr. Hosny so nicely set me up for, the pathophysiology of lupus is really complex, but there does seem to be genetic drivers of this disease. So we know that at least in some ways lupus is heritable. So the twin concordance is anywhere from 24 to 56%, depending on the study and the population. And people with lupus, siblings of people with lupus, have 26 times the risk of developing lupus than the general population. Now, if we think about who's been studied so far, it's actually been genome-wide association studies, so studies using SNP arrays, so looking at common variants on a chip in adult patients. And whole exome or whole genome study looking more comprehensively at genetic drivers has been done in some patients, but actually usually those patients are childhood onset patients, but they're a really small subset of childhood onset patients. So there have been patients that maybe have age of onset really, really early, like 4 or 5 years old, and somebody says this is pretty unusual, we should sequence them, or they just have a really strange phenotype. So even amongst all the childhood onset lupus patients, those of whom we know the genetic architecture is pretty limited. Now, if you think about drivers of disease, if you look at this chart here, common variants, which we pick up on GWAS studies, usually have really small effect size. So they're passed down through many generations. They don't cause too much damage, so the people who have these variants kind of survive to pass on these genes to further generations. So the thought is that in adult onset patients, there's many of these common variants kind of acting together with maybe some environmental factors. And then if you think about a rare variant, they're usually very damaging, very critical, a very large effect size, but not common in the population because they're so damaging, they're not usually passed on to future generations. Now, the thought is in childhood onset lupus, the very early patients have kind of a monogenic picture, so one of those large damaging variants. And as they get older and get up into the adolescent stage, they probably have more of a polygenic picture like their adult counterparts. And so this is kind of the spectrum of disease, but actually this has never been studied for just the reason that I talked about earlier, that we have GWAS patient studies on adults, very few genome-wide association studies on kids to look at the common variants, and those that we've studied in the early with whole exome or whole genome sequencing have been a very small subset of patients. So this is the paradigm that exists, but it's actually a hypothesis that hasn't been tested. And then there's just kind of a global disparity in genomic research that I'd like to highlight. So this is not isolated to lupus. This is just kind of how the field of genetics has evolved. So if you look at who's been studied, most of the studies in lupus have been GWAS, as I mentioned, but about 80% of all genetic studies have been done in patients of lupus of European ancestry. And only about 20% is all other ancestries total to date. And then if you look at the population here, so 10% of those are Asian, but 8% of those are East Asian. So anybody who's not East Asian, it's only 2% of anybody that's ever been studied. So we actually have only studied lupus and common variants in adults of European ancestry who are actually statistically the least likely patients to have severe disease. So if you think about who gets severe lupus, it's usually people who are non-white in many different places around the world, and then children. And those are the patients that we've genetically studied the least, even though they're most likely with an early onset and severe disease phenotype to be the most likely population with a genetic driver. In the adult GWAS that has been done, there have been about 100 risk loci developed. It's important to note that when you do a GWAS, it's really important to control for the background population. So a variant is only common if you know what the genetic landscape of those patients are. So you have to know the allele frequency of a variant in that population. You can't use a Pakistani population with a European allele frequencies because they will be different based on genetic populations. So this study is a trans-ancestral mapping from the US. So they did a study in US, African American, Hispanic, and then did a meta-analysis, and you can see that the SNPs that they find are different based on population. I bring this up just to note that there have been some really interesting studies when you take a deep dive into diverse populations. So the APOL1 story is kind of my favorite study, is my favorite one that highlights this. So APOL1 risk allele is common in African ancestry, but rare in European ancestry. So it didn't pop up on a general study of people of European ancestry because it's not a common variant in that population. It is actually common in people of African ancestry because it's protective against African sleeping sickness, so that evolved over time to be beneficial for the population. But it is, when they did an African American GWAS in the US, they found that it was associated with lupus nephritis. And then Dr. Blazer actually did another study looking at the population in Ghana, because most of our African American population is from West Africa, and found that APOL1 was associated with damage accrual in lupus patients despite no difference in disease activity, and was associated with a 14-fold risk for mortality. So this is just one study that really highlights the importance of studying diverse populations, because what is true genetically for genetic drivers in Europeans may not be true in other populations. Another way to kind of look at the burden of common variants in a population is to look at a polygenic risk score. So if you take all of the risk loci known for lupus and you sum them up, you can get kind of the burden of common variants in a person or a population. And it incorporates both like the number of lupus risk SNPs that you might have and the effect size of those variants. And so most of the GWAS studies that have been done in children are actually studies that are looking at polygenic risk scores. So a study by Earl Silverman's group out of Toronto showed that if you look at childhood and adult onset lupus patients, they studied common variants in both of these populations, that the burden of common variants as you associated with age is different. So they first looked at the SNPs outside of the HLA region and found that it was negatively associated with age of onset, but in the HLA region, it was positively associated with age of onset. And then Linda Haraki's group, also in Toronto, looked at the adult and childhood onset lupus patients to see if they had a differing burden of common variants, and found that in most populations, so they did different ancestries, admixed, African, East Asian, European, and South Asian, and found that although there was a trend for childhood onset patients to have a higher burden of common variants than adults, it wasn't statistically significant except in the South Asian population. So maybe a larger study would show that childhood onset patients overall have a higher burden, but this was like a little bit surprising because, again, it was thought that childhood onset patients were more likely to have those rare variants and adults were more likely to have the common variants. They did also stratify by ancestry and look at the odds ratio for developing certain clinical phenotypes, and found that a higher risk of common variants was more associated with developing lupus nephritis in almost every population that they studied. So over time, as we collect more and more data and look at risk SNPs in more populations, we may be able to use some of these polygenic risk scores to predict clinical phenotypes. So whole genome analysis includes rare and common variants and allows not only for us to look at known rare variants, but allows for novel rare variant discovery. The best way to do that is actually with family studies. So whole genome data gives you a whole lot of data. So if you have a child and their unaffected parents, you can really assess novel rare variants that way. And you can also look cohort-wise. So if you find a rare variant in a immune regulating gene in one patient, and then you find it in another unrelated patient, and both of those patients have lupus, it might be more likely to be causing disease. So this is a really exciting field as kind of whole genome sequencing is being practiced more often. I feel like, by tomorrow, the genes that I talk to you about in this talk will be outdated because there's something new coming along every day. But these are some of the variants and pathways that have been studied as causes of monogenic lupus. And if you just look at, we're starting to do larger cohort studies of patients. So this is a study that came out of France, and they didn't do whole genome sequencing. They actually did a panel of immune-regulated genes. But they did 174 childhood onset lupus patients, so a pretty big number, and then 574 non-lupus controls. And they just wanted to see if there was a difference in the number of rare and damaging variants in these patients. And they found in immune-regulated genes, about 1 in 4 lupus probands had a rare predicted damaging variant, versus just 5% of controls. So again, this is evidence that childhood onset lupus patients are probably an enriched population for genetic drivers of disease. This study out of Texas kind of confirms that. So in theirs, they did whole exome sequencing on 48 patients, and over 10% of those had they were able to solve those cases, so an absolutely determined monogenic case of early onset lupus. And they found a couple different pathways, RAS-associated autoimmunity, DNase 1L3. And if they looked in patients who were really young, it was 60% of those patients. So again, kind of saying that there's certain populations that are more likely to have monogenic etiology. And then kind of thinking about the pathways, I don't really have time to go through all of these today because there's a lot of different variants, and it would maybe take me an hour to go through all of them. But if we think about just the different pathways, there's tolerance, there's either loss or gain of function in some of the nucleases, nucleic acid sensing, and complement deficiency. So complement deficiency is the oldest genetic association. It's been known, there's genetic drivers of lupus for more than 50 years. It was discovered in the 1970s. The different components of the early complement pathway have different risks for lupus. So if you have a C1q, it's about 90% of those patients will have a lupus phenotype, and then going down the line, C4 is about 60%, and then C2 is small, about 10 to 15%. And the way that this develops into a lupus phenotype is multiple pathways. You can have disordered apoptotic clearance and impaired immune cell processing. And then the other kind of big, the bigger field is or more broad kind of pathways are nucleic acid sensing. So there are extracellular nucleases like DNase-1 and DNase-1L3 that can have a loss of function mutation where they don't break down the DNA, and then that DNA kind of triggers some of the sensing pathways to create an interferon signature. There's intracellular nucleases such as MDA5 and RIG-I through the RNA sensing pathway. Intracellular DNases such as TREX1 and DNase2, and then that can be gain of function or loss of function, and then these pathways kind of go through this the signaling pathway to trigger an interferon signature and a lupus phenotype. So this is just the known pathways. There might be more coming down the line, but I will have to say that when we study childhood onset lupus patients, we don't only understand childhood onset lupus. Once they found that TREX1 was associated with Aicardi-Gouttiere and Chilblain lupus and early onset lupus patients, another study kind of looked at targeted sequencing of TREX1 in adult onset patients, and a good number of those patients had heterozygous mutations in TREX1. So understanding early onset lupus patients actually helps us understand pathophysiology of all patients. And I think the TREX1 story really was an essential part of the story for us to understand the role of interferon signatures in lupus as well, which has been a really rich area of study and might be therapeutic target. And as mentioned earlier, I think the genomic studies are really just the first point, because once we find the genomic variant, we have to understand, what transcripts are those making. So we have to understand transcriptomics. We have to understand how those genetic variants are making altered proteins and how those pathways and mechanisms are leading to a lupus phenotype. We also have to understand, which immune cells are expressing those genes. They're not uniformly expressed or important in all cells, so those studies will kind of be the next steps down the pathway of these genetic studies. And I think collaboration is really important. So, as I mentioned earlier in the talk, we have really only studied the genetics of lupus in European ancestry, and it's my personal mission to advance the research, the genomic research in many, many more populations, because I think the more that we understand about lupus anywhere, the more we understand about lupus everywhere, and the way forward is through collaboration. So I hope through these studies we can advance the understanding of the genetic architecture of childhood onset lupus in diverse populations, and hopefully those studies will take us from a broad umbrella of this clinically heterogeneous phenotype of lupus to precise molecular definitions of disease that will help us target therapy and improve disease outcomes. Thank you.